Welcome to the Explainer. So today, we're undertaking this massive physiological journey. If you're gearing up for a huge medical exam, you know the pressure is on. But hey, instead of stressing out, let's just marvel at how these systems actually work. We're going to maximize our understanding of two vital networks in record time. Let's dive in.
Think about cricket legends for a second. Guys like Sachin Tendulkar or Virat Kohli. When you're watching a gripping match, you know for a fact that the last over means absolutely everything. The entire game changes right there. Some players completely turn the tide with a hat-trick of sixes. Your final days of preparation are exactly like that last over. It really doesn't matter what your mock scores have been up to this point. Focused, unyielding effort right now changes the game. No more underestimating yourself, okay? We're going to make this last over count. All right, here's our game plan for today. We're going over the final revision push, the human respiratory system, lung volumes and capacities, transport of gases, blood composition and cells, and finally, blood groups and coagulation. Let's get to it. Okay, Section 1, the human respiratory system. Let's look at the anatomy of breathing.
Now, before we get into human anatomy, we really got to distinguish between respiration, the cellular oxidation of food, and breathing, which is basically just the physical machinery. And it's actually fascinating how this machinery evolved. Earthworms, they just rely on a simple moist cuticle, basically their skin, for gas exchange. Insects, like cockroaches, use this whole network of tracheal tubes. And as we move into aquatic environments, fish use these specialized vascularized structures called gills. It's a perfect example of how organisms shifted from simple diffusion across the body surface to highly specialized respiratory organs as they got more complex.
So what does the human pathway look like? Well, it's an incredibly efficient, highly specialized conducting network. Air comes in through the nostrils, moving right into the nasal chamber. Then it hits the pharynx, which, remember, is the common passage for both food and air, before it reaches the larynx, our cartilaginous voice box. From there, it travels down the trachea, or windpipe, splitting into bronchi, then increasingly smaller bronchioles, until it finally hits the actual site of gas exchange, the alveola. And honestly, the destination itself is what's really interesting. The alveoli are the critical respiratory endpoints. Picture these incredibly thin, highly vascularized little bags. This is the exact spot where the magic happens, where oxygen from the air literally swaps places with the carbon dioxide waste from your blood through simple diffusion. Everything before this was just the conducting part, simply meant to filter, humidify, and warm the air. Moving right along to Section 2, lung volumes and capacities. Time to master the metrics. To really understand our lungs, we look at metrics measured by an instrument called a spirometer. Let's anchor our first big number, 500 milliliters. That's your tidal volume, or TV. It's really just the normal volume of air you inhale or exhale with every single relaxed breath. Like right now, as you're listening to me, you're quietly moving 500 mils of air in and out. But how do we physically do that? It's all basic physics happening right inside your chest. Specifically, the inverse relationship between volume and pressure. During inspiration, your diaphragm and external intercostal muscles contract. This expands your thoracic cavity, boosting the volume. Because the volume goes up, the pressure inside your lungs actually drops below the atmospheric pressure outside, creating this negative pressure vacuum that just pulls the air right in. Expiration is totally the reverse. It's mostly just a passive relaxation of those muscles. Volume shrinks, pressure goes up, and air is naturally pushed out. Now, obviously, we can push our lungs way past that normal 500 ml limit. Vital capacity is the absolute max amount of air you can inhale after you've forcefully pushed a breath completely out. Total lung capacity is the absolute maximum air your lungs can physically hold. But here's a really surprising fact. Even if you push out every ounce of air with all your might, you've still got a residual volume of about 1,100 to 1,200 milliliters left over. Why does this matter? Because it keeps your lungs perpetually inflated. It literally prevents them from collapsing. And since it never leaves the lungs, a spirometer can't even measure it. All right, Section 3. Transport of gases. Let's talk about oxygen and CO2 in transit. The transport story for oxygen is super fascinating. A massive 97% of the oxygen in your body relies entirely on hemoglobin inside your red blood cells. Think of hemoglobin as this dedicated transit bus with 4 specific seats for oxygen molecules. Only a tiny fraction, like 3%, is just dissolved directly in the blood plasma. So what decides if oxygen gets on or off that bus? That's all determined by the oxyhemoglobin dissociation curve. A shift right means dissociation. The hemoglobin drops off the oxygen. This happens at your tissues where carbon dioxide is high, acid is high, and temperature is high. Conversely, a shift left is association, grabbing oxygen, which happens in the lungs where oxygen is plentiful and CO2 is low. Make sure you remember this vital stat. Every 100 milliliters of oxygenated blood delivers exactly 5 milliliters of oxygen to your tissues. Oh, and behind the scenes, your respiratory rhythm is constantly moderated by neural controls, specifically the pneumataxic center located right in the pons of your brain. Now carbon dioxide's commute looks totally different. Surprisingly, 70% of CO2 is actually transported as bicarbonate ions in the plasma. This incredible conversion happens fast inside the red blood cells, facilitated by a supercharged enzyme called carbonic anhydrase. About 20 to 25% travels bound to hemoglobin, that's called carbaminohemoglobin, and roughly 7 to 8% is just dissolved in the plasma, mostly because CO2 is way more soluble in water than oxygen is. Next up, Section 4, blood composition, the body's fluid connective tissue. Blood is actually quite unique because it's the only connective tissue that completely lacks structural fibers. If you spin a tube of blood down, it separates into two main parts. 55% of blood volume is plasma, that straw-colored viscous fluid that is mostly water but absolutely packed with critical proteins. For instance, it's got fibrinogen, which is essential for clotting, and albumins, which maintain your osmotic balance. The other 45% consists of the formed elements, which is really just a fancy clinical term for your red blood cells, white blood cells, and platelets. Let's meet the body's internal defense squad, the white blood cells, or leukocytes. Neutrophils are by far the most abundant, making up 60 to 65%. They are your primary phagocytic cells. Think of them as the frontline soldiers literally eating up foreign invaders. On the totally opposite end, basophils are the rarest, releasing histamine to trigger inflammation. Eosinophils are the ones that spike during allergic reactions. Lymphocytes, those are your B and T cells, driving the really major immune responses. And finally, monocytes, much like neutrophils, also serve a phagocytic function, just gobbling up cellular debris. Finally, Section 5, blood groups and coagulation. Let's dig into some core survival mechanics. Your body's ability to recognize its own blood is a literal survival mechanism, and this explains the whole logic behind blood typing. Your blood type is dictated by the presence or absence of specific surface antigens on your red blood cells. O negative is the universal donor. Why? Because it literally has 0 A, B, or Rh antigens on its cell surface. That means anyone's immune system will accept it without sounding the alarm. Conversely, AB positive is the universal recipient. It has no antibodies in the plasma against A, B, or Rh, meaning it can peacefully receive any blood type without attacking it. This antigen logic also brings us to a fascinating immunological puzzle known as erythroblastosis vitalis. Basically, this happens when an Rh-negative mom has an Rh-positive baby. During the first delivery, the mom's immune system gets exposed to the positive blood and builds antibodies against it. If she gets pregnant with a second Rh-positive baby, those antibodies can actually cross the placenta and attack the fetus's blood. Fortunately, modern medicine gives us an incredible triumph here. The ROGAM vaccine. It's administered to the mother to totally neutralize this threat and protect future pregnancies. Okay, what happens when you actually suffer a trauma? You don't just bleed endlessly, right? That's thanks to a cascading waterfall effect called coagulation. When injury occurs, the tissues release thromboplastin. This kicks off a fast-paced enzyme cascade where thrombokinase activates inactive prothrombin into its active form, thrombin. Then thrombin works its magic by converting soluble fibrinogen into insoluble fibrin threads. These threads form a literal mesh that traps blood cells and creates the clot. And the absolute MVP requiring all of this to function properly? Calcium ions. Without calcium, this whole waterfall dries up. So, are you ready to dominate this exam? We have just sprinted through an immense amount of high-yield biology, from the deepest depths of your alveoli to the cascading proteins of your blood plasma. You have the structural understanding, and you've got the logic down. Now it's just about putting all of these fascinating systems together as you head into your final push. Let this be your absolute mantra for your preparation. Yes, I can do it, and I will do it. But beyond the exam, I really want you to ask yourself this. If our bodies flawlessly execute these incredibly complex cascades of survival every single second without us even trying, what other biological miracles are happening inside you right now? Thank you so much for joining me on The Explainer. Keep learning, keep pushing, and I'll catch you in the next one.